get started, I just want to mention that I'm going to be talking about some drug that I developed that Jameen mentioned, and I have a conflict of interest to disclose. We have a company at Columbia Patents, and I have a commercial interest in this. What I'd like to talk about today is this molecule, the Ryanodine Receptor. I realized last night in between shots of whiskey that I've been working on this almost 30 years. I'm very slow. It takes me a long time to get things done. But I originally cloned this in 1989, and then in 2015 was able to see what it looks like finally. And the Ryanodine receptor is the largest known ion channel. The enormous part of the channel sits in the cytoplasm here and serves as a scaffold for regulatory proteins, and this is the transmembrane part in the sarcoplasmic or endoplasmic reticulum in almost every type of cell. For 10 years, I spent trying to crystallize this molecule in collaboration with Wayne Hendrickson at Columbia, and we now know that this flexibility here was probably the reason that the resolution of our crystals never got better than 7 angstroms. And then with the revolution of cryo-EM in 2014, we were very quickly, in about 6 months, able to solve the high-resolution structure that I will talk about more today. And then I'll also tell you some stories about how the Ryanodine receptor becomes leaky, either with mutations or stress, and when the calcium leaks out, it can cause or contribute to a number of human diseases. So Ryanodine receptors, as I mentioned, are homotetrameric ion channels, the largest known channels. Each monomer is 560,000 daltons, and the mass of the entire complex is over 3 million daltons. These little blue molecules here are FKBP or calstabin, which I discovered are part of the channel. It is a stabilizing subunit that stabilizes the closed state of the channel and prevents a leak. The story I'll tell you today is that when the channel under stress becomes oxidized or phosphorylated, or due to genetic mutations, a calstabin no longer binds tightly and the channel leaks. But the channel is required for fundamental processes, for example, neurotransmitter release in the brain, cardiac excitation-contraction coupling, as well as in skeletal muscle, and even in the pancreas and pancreatic beta cells. And this, I know, is a review for all of you, but the Ryanodine receptor is a macromolecular signaling complex, and not only does it bind the stabilizing subunit calstabin, but also kinases, phosphatases, phosphodiesterases that are targeted to the channel via targeting proteins, as shown here. And when the membrane is depolarized, the voltage-gated calcium channel in the heart allows a small amount of calcium to enter, or in skeletal muscle, it interacts directly with Ryanodine receptor, which opens, releases a much larger amount of calcium, which binds to troponin C, allowing shortening of the sarcomere. And this is the basis for muscle contraction. And then that same calcium gets pumped back into the SR via the SR pump, ATPase. And so this process is called excitation-contraction coupling, because it converts a action potential electrical energy to mechanical energy by the rise and fall of calcium. And the whole system is also regulated by extracellular stresses. In this case, during exercise, for example, catecholamines activate the canonical pKa pathway, and all 3 of these critical EC coupling components are phosphorylated and activated by pKa. So, as I mentioned in the end of 2014, the resolution of single particle cryogenic electron microscopy was greatly improved and up to near atomic resolution. And so we took advantage of this and collaborated with Joachim Frank at Columbia. And very quickly, what one does is take a protein sample. In this case, we had highly purified Ryanodine receptor type 1 from rabbit skeletal muscle, which we had been purifying for 20, 30 years. And so we had really great protein. You put it on this gold or carbon grid, flash freeze it in liquid ethane at minus 170 degrees, making a very thin sheet of ice that has your protein of interest randomly oriented, image it with electron microscope, and generate 3D images based on the random orientation of your protein. And then from this, you develop a model as shown here. And we currently have the entire molecule solved at 3.1 angstroms. And I'll show you some data on that. So in late 2014, we published the first structure. And what we found was of note that the Ryanodine receptor had the structure of a 6-transmembrane cation channel, which was not previously known, and the transmembrane segment shown here, an extension of the S6 transmembrane segment interacting intimately with the carboxy terminal domain, which in turn interacted with this enormous cytoplasmic domain that contained binding sites for the ligands and proteins that regulate the pore. And for example, here in yellow is shown the calstabin that I talked to you about. Fortuitously, during this experiment, there was a class of particles that had only 3 of the monomers, and so we were able to rigorously define the borders of each of the monomers, and shown here is one of the 4 monomers in color. It turned out that these sequences in the cytoplasmic domain were a unique structure that was formed by alpha helical repeats and has a canonical name of armadillo repeats, very flexible structures, and I'll show you why that's important. Now, I know a lot of you work on the potassium, sodium, calcium channels and trip channels, and this was really quite a stunning observation that the structure of the pore of the Ryanodine receptor, which is not a voltage-gated channel, it has a much larger conductance of 100 picosiemens for calcium, but as you can see, structurally almost identical to the potassium, sodium, and trip channels. Yet the Ryanodine receptor in gray lacks a selectivity filter and only has 11 to 1 selectivity for cations over anions, and actually passes sodium and potassium better than calcium. So the conductance for sodium, for example, is about 700 picosiemens. We also observed a large cluster of negatively charged residues at the mouth of the channel in the sarcoplasmic reticulum lumen, suggesting that calcium could be a very high concentration in this area, attracted by all these negative charges. A very surprising finding was that the diameter, as I'll show you, of the open state of the channel was only about 11 angstroms, meaning that it could only pass a single hydrated calcium at a time. So the calcium has to line up in single file. A single hydrated calcium, as you know, is about 8.5 angstroms. With the very large conductance of the channel, we had expected a much larger aperture. And then another feature was this, I mentioned this extension of S6 from the pore transmembrane region up into the cytoplasm, and that was characterized by a glycine hinge that causes about a 24-degree bend in the channel pore, which I'll show you is important for gating of the channel. So we were able to define discrete regions of the cytoplasmic domain as shown here, and one of the features is this enormous bridging solenoid shown in green, and interestingly, the closely related IP3 receptor, which is smaller than the Ryanodine receptor, lacks this bridging solenoid. Also an N-terminal, C-terminal solenoids, and they're shown here, and then the pore of the channel. And as I mentioned, in yellow, you can see this extension, once again, of the S6 domain going up into this region of the cytoplasmic domain. And here you see what we refer to as a pseudo-voltage sensor, because it has all of the transmembrane components that you're very familiar with, S1 through S6, but the S4 transmembrane segment lacks the positive charges that one finds in voltage-gated channels. So either during evolution, the ryanodine receptor lost these charges or never obtained them. And so this is remarkably similar structurally to the channels that all of you work on, but again, the ryanodine receptor is not voltage sensitive. And through the last 2 years, we've been able to consistently improve the resolution of the entire structure now, and we're currently at 3.1 angstroms, and we've solved the closed and open states, as you can see here. We were able to force the channel into an open state by adding 3 activators, calcium, ATP, and caffeine, and we got a nice set of particles that were clearly open, as shown here. And you can see the remarkable resolution that we have now with all of the side chains very clearly identifiable. So just as a byproduct, does anybody know why the ryanodine receptor is called the ryanodine receptor? Huh? And what is ryanodine? He's a ringer. So this is a plant that grows in South America, and this is a native in South America with a blow dart putting some ryanodine on the tip of the blow dart. And when ryanodine gets into the body, it locks the ryanodine receptor in a characteristic half-open state. Calcium floods out in your heart and skeletal muscle, and your heart turns to stone and your diaphragm can't move. Why does the plant have ryanodine in it? Well, it's a wonderful insecticide. When an insect lands on this plant and takes a bite out of the leaf, it also gets essentially paralyzed and falls off. And so the name came from this plant, Ryanodine speciosa. And we can actually see ryanodine now binding in the pore of the channel shown here. And we're able to define the ryanodine binding site. Ryanodine has characteristically 2 effects on the channel. As I mentioned, there's a high affinity effect when there's low concentration of ryanodine. It locks the channel in a half-open state, shown here, and you do this at the end of every bilayer experiment to make sure you're studying ryanodine receptor. And at higher concentration, it blocks the channel. What we were able to observe is that at low concentration, there would be a single molecule of ryanodine in the pore, partially blocking the pore, accounting for this partial conductance state. By the way, here are the closed channels, and this is the full open. And as we raise the concentration of ryanodine, we could force 3 additional molecules into the pore and completely block it. Okay, so typically, ryanodine receptors are known to be calcium-activated channels, but if you add an activating concentration of calcium to the cytosolic side of the channel in a planar lipid bilayer, you get an open probability of 10 to 20%. However, if you then add ATP and caffeine, the combination of all 3 results in a very high open probability, about 90%. So we took advantage of this, as I had mentioned, to solve the open state of the channel. But we also were able to identify the binding sites for these 3 critical ligands, calcium, caffeine, and ATP. And remarkably, they're all clustered in the same region, which is a site that is between a flexible domain in the cytoplasmic part of the channel and the pore. And it appears that what the other ligands are doing is rigidifying this flexible interface here so that when calcium binds, activation by calcium is made more efficient and results in opening of the pore. And so you can see this in these movies where when you have EGTA, so low calcium, you add only calcium 30 micromolar, draw your attention to the pore here, and there's very little, if any, motion in the pore. So the 30 micromolar calcium is not causing very much activation of the pore. However, when you add the caffeine and ATP and the calcium, now you can see an opening of the pore when you add these activating ligands, and you can see much higher open probability. So one of the things that we were very interested in doing was taking advantage of the high-resolution structure to try to better understand how mutations in RyR1 in skeletal muscle and RyR2 in the heart cause important human diseases. So in RyR1, there's a disorder called malignant hyperthermia syndrome, which occurs when you go for surgery and the doctor puts you to sleep with inhaled gas, halothane, usually, and that causes a leak in the Ryanodine receptor, and you essentially burn up ATP, trying to pump the calcium back in. Muscle cells necrose, you release potassium, and it's a lethal disease that can be prevented with a drug called dantrolene. Central core disease is an old pathologic term that has now been recategorized as RyR1 myopathy, and it's a form of progressive muscular dystrophy with muscle weakness I'll talk more about later. CPVT and RyR2, or catecholaminergic polymorphic ventricular tachycardia, is a form of exercise-induced sudden death that I'll also talk about in a moment. And it's a devastating disorder. It's probably the most lethal known ion channel mutation, and it occurs in children with normal cardiograms, normal hearts anatomically, and when they start playing sports at school, for example, the heart rate gets elevated. It triggers ventricular tachycardia and fibrillation and sudden death. And we've mapped all of the mutations, and in fact, we've mapped almost 1,000 mutations in RyR1, and we're working on RyR2 now, up to several hundred. And they do cluster in certain regions, as you can see from these dots, and we're trying to better understand what they are doing. So we've looked at each one of the ligand binding sites, and here's the calcium site. And these red residues are human disease-associated mutations. And you can see them clustering around where the calcium binds. One of the first things we wanted to do was to confirm that this site is actually a calcium binding site. So we introduced artificial mutations as shown here, and we're able to measure the open probability of the channel, or in this case, ryanodine binding. And what we found was quite surprising and unexpected, is that when we mutated the calcium binding site, as shown with these missense mutations, we not only lost the high affinity activation of the channel, which occurs at low concentration of calcium in the micromolar range, we also lost the low affinity deactivation of the channel, which typically occurs in a millimolar range. This would suggest that this single site is somehow both a activating and a deactivating site for calcium, and we are now trying to better understand that. Similarly with ATP, again, draw your attention to these red residues, and they are all human disease-associated residues. And by the way, one of the challenges here is we don't know whether any of these are disease-causing. And I regularly get emails from physicians around the world who say, my patient has such and such a mutation, what should I do? And for 99.999% of these, we can't answer that question yet. And that's one of the challenges we're undertaking. One of the things that's very interesting about this particular site for ATP, so here is the head of the ATP, the triphosphate tail. There's a mutation in one of the patients that is a threonine to methionine mutation here, and you can see when you put in a methionine, there's a steric hindrance with the adenine ring of the ATP. And when we mutate that residue shown here, we get a dramatic decrease in ATP binding, but mutations in the residues that interact with the triphosphate tail do not reduce ATP binding. However, they do reduce activation of the channel by ATP. And finally, there's a caffeine binding site, and again, these red residues are disease-associated human mutations. And caffeine, as you know, is a methylxanthine. It's not a naturally occurring molecule in mammals. It's a plant product. The closest molecule to caffeine that I'm aware of in mammals would be xanthine, which is in the uric acid pathway. And we are currently trying to understand what actually occupies this site in vivo. Calstabin is the stabilizing, yeah. Right. So yeah, I'm just showing mutations that are known to occur in patients that are near the caffeine site. We don't know whether they're causing disease. We have to go through and make those mutations, see how they affect the channel, and do a lot more work on that. So again, the calstabin is shown binding here with the higher resolution. We've been able to identify all of the interacting residues and the hydrogen bonds. And calstabin is knocked off the channel when there's nitrosylation, and we've identified a cysteine residue involved in that, and then also oxidation and phosphorylation. And again, getting back to one of the patients that we've been working up, there is a mutation in this valine that converts it to an isoleucine, and that occurs in one of the regions where the calstabin interacts with the channel, and the patient does exhibit leaky channels and decreased calstabin binding. Another interesting molecule that regulates ryanodine receptor is calmodulin, and there are also disease-associated mutations in the calmodulin binding site. And calmodulin adopts a very unusual structure when it's bound to ryanodine receptor, with the N and C lobes stretched out, and doesn't undergo the classic changes in structure when calcium binds. But the binding of calmodulin dramatically stabilizes the channel and has allowed us to improve the resolution. So over the last 20 years or so, we have defined the effect of leaky ryanodine receptors in these different organs, as Yamin mentioned. And so leaky ryanodine receptors in the brain decrease the seizure threshold and also cause cognitive dysfunction in a murine model of post-traumatic stress disorder, as well as in Alzheimer's. And in unpublished data, we are finding similar effects in mouse models of Huntington's disease. In the heart, leaky ryanodine receptors can contribute to heart failure progression and arrhythmias. In skeletal muscle, they play a role in muscle weakness and muscular dystrophy. There's a term called sarcopenia, which is better thought of as age-dependent loss of muscle function. About 50% of individuals, when they reach their 80s, develop muscle weakness. And we've shown that leaky ryanodine receptors contribute to that. One of the major paraneoplastic syndromes in patients with cancer, especially metastatic, is cancer-associated muscle weakness. And in collaboration with Teresa Geis at Indiana University, we've also shown that leaky ryanodine receptors play a role there due to oxidation by activation of the TGF-beta pathway. In pancreatic beta cells, leaky ryanodine receptors contribute to mitochondrial dysfunction, decreased ATP, and impaired insulin secretion. And we were able to make mouse models of leaky ryanodine receptors associated with the exercise-induced sudden death, CPVT, and show that they all had glucose intolerance and decreased insulin secretion. We then contacted physicians taking care of 20 patients with these mutations in RYR2, young, healthy people, no suspected metabolic abnormalities, and they all had glucose intolerance and decreased insulin secretion. So one of the challenges that these studies presented a number of years ago was conceptually, if one could fix the leak in the ryanodine receptor, you could potentially come up with novel therapy for heart disease, muscle, brain, and so on. And in looking through the literature, I found a molecule called JTV519 made by Japan Tobacco when they were synthesizing diltiazem, and it had been put aside. And then after reading my papers, a group at Yamaguchi University took the JTV519, treated a dog model of heart failure, and following on our reports showed that it fixed the leak in the Ryanodine receptor, rebound calstabin, and improved the heart failure. And so this was like a dream come true. And but that JTV519 is insoluble and has potent hERG block. So instead, I asked the chemists at Columbia to make a number of compounds, one of which is shown here, and this is called S107 that you'll hear me mention. And then another one of these, which we generically call RyR-Cals, is in clinical trials that I'll mention as well. So we're very interested to know where the RyR-Cals bind on the Ryanodine receptor. And this is a single experiment that we've done. This is a difference map showing a density here that corresponds, we believe, to the RyR-Cal. And quite interestingly, it's in the caffeine binding site. So again, we need to repeat more experiments. I just show this. I would never publish this because it's really just an N of 1, but we're able to see molecules of this size, and we're using cryo-EM to try to define the drug binding site. So I've mentioned CPVT. It's a very rare form of sudden cardiac death, and the baseline electrocardiogram is normal. When you see horrible scenes like this of young, healthy athletes dropping dead suddenly during sports, is one of the causes or mutations in the Ryanodine receptor. The heart function and structure are normal. As I mentioned, it presents as sudden death in young children and young adults, and it's caused by mutations in RYR2. And as the heart rate increases, you develop these ugly-looking cardiograms, and then this is what's known as a polymorphic ventricular tachycardia, and eventually goes to something called ventricular fibrillation and death. So patients now, it's become fairly well understood this is a possibility, and doctors now are ordering genetic testing, including sequencing Ryanodine receptors, and we're collecting a lot of data about this, but we don't know what to do with a lot of it. And as time goes by, we're continuing to collect more and more mutations, and these large purple balls are clusters of mutations, and again, they form in regions between domains of the Ryanodine receptor where there's particular flexibility, and the mutations increase the flexibility and destabilize the channel. So a number of years ago, we made mutations in the human recombinant Ryanodine receptor, and when you mimic exercise by adding catecholamines or PKA phosphorylate in the channels, they have increased open probability as shown here when you add PKA as compared to a wild-type channel at non-activating calcium concentration, and decreased binding affinity to the calstabin. Increased calcium sparks, and knock-in mice develop the same kind of abnormal rhythms and sudden death as in patients. When we treated these models of human CPVT with that RyR-Cal S107, as I mentioned, which acts by preventing the loss of the stabilizing subunit calstabin, we're able to decrease ventricular tachycardia and sudden cardiac death in this mouse model. I want to now tell you a story about an individual who has what is called RyR1 myopathy. This used to be called central core disease because, as you can see, the center of the myofibrils is very light colored. And the typical presentation, as shown in this cartoon, is proximal muscle weakness. So the individual has to push himself up to stand. So this patient I met about 4 years ago, and was having a discussion with him about he eventually started a foundation and asked me to help with that. And when he was sitting there talking to me, you would almost think he was normal, except at the end of the meeting, his wife came and hooked her arms under his armpits and had to pull him up to a standing position. At that time, he told me that he had muscle weakness in his lower extremities, his quadriceps, but his arms were normal. He's a radiologist and very functional guy in his 40s with 2 kids. And so we had done a genetic diagnosis and found that he was a compound heterozygote, having inherited that threonine to methionine mutation from his father. And from his mother, he had a leucine deletion and an arginine to cysteine mutation. And so he came to me several times and said, Andy, would you arrange to have a biopsy done of my muscle? And I refused. I said, what I always teach the medical students at Columbia is never order a test on a patient unless you know what you're going to do with the results and that it can help the patient. And I said, we don't really have anything to offer, and I don't know what we could benefit from. But he was very, very persistent, and we ended up doing that biopsy, and we also obtained 21 biopsies from around the world from docs. One of the nice things about working on Ryanodine Receptor is we just need frozen muscle. We don't need live tissue, so we can get all the information we need, including channel activity. So what we found was we analyzed his muscle for what we call the biochemical signature of leaky channels, and that includes oxidation of the channel and nitrosylation. And what we found was that in the quadriceps, but not in his deltoid, so the affected muscle was the quadriceps, there was oxidation, nitrosylation, and depletion of the stabilizing subunit from the channel. But we really didn't see that in the deltoid or in normal controls. And we then took his muscle lysate and treated it with the RyR-cal ex vivo, and were able to rebind the calstabin to his muscle. And we did that with 2 different RyR-cals, this one and this one, ARM36, which is also in clinical development. So we then wanted to try to understand which of the mutations would be causing the leak, and we made all the different combinations in the recombinant human Ryanodine receptor, and only the mother's mutation, the leucine deletion and arginine to cysteine missense mutation, caused increased open probability of the channel under non-activating conditions. And the other ones individually did not. However, on further study, we found that the T47 08 to methionine mutation, as I showed you, impaired ATP binding to the channel. So we then treated the lysate, as I mentioned, with the RyR-cal, and we looked at the single channel activity, and we're now using one of the RyR-cals called ARM210, and this is entering phase 2 clinical trials that I'll tell you about. And again, here there was a leaky channel phenotype, and when we treated the muscle with the RyR-cal, that decreased the open probability, and in a microsomal leak assay also decreased the leak in the channel. This is an assay where we make microsomes. We put a calcium-sensitive dye outside. We load the microsome with calcium by adding ATP. The Circa pumps calcium in, and then we block Circa with epsigargan and measure passive leak. And we know this is through the Ryanodine receptor because at the end of the experiment we block it with Ryanodine. So just to finish up, what I've told you is that we've developed a new class of drugs. I didn't show you the data, but it's all published showing that we can prevent heart failure progression. The drugs are anti-arrhythmic and improve exercise capacity in animal models. And it occurs by the binding of the compound to the channel, the binding of the stabilizing subunit, and that restores the normal function of the channel. In early clinical trials in patients with heart failure, the RyCals improved ejection fraction, reduced the left ventricular size, and decreased a marker for heart failure, brain natriuretic peptide. And we are now working with a second-generation RyCal, ARM210, that is more potent and has completed safety trials, phase 1. And starting in January, we're going to be taking those same patients 21 patients that we treated their muscle lysate with the RyCal, and we're able to fix the leak. We're now going to give them the drug orally for 1 month and rebiopsy them and see if we can fix their leaky channels. We're also starting a trial in Duchenne muscular dystrophy, another form of myopathy. So I just want to finish up with this concept of functional genomics. There, as you know, is a huge interest nationwide and enormous amounts of money being pumped into precision medicine, and that means sequencing patients, exomes, 1 million exomes, and so on. And that's going to generate a huge amount of sequence data, and we're not going to know what to do with most of it. So every one of you in this room has a number of mutations, and we have no idea, probably all of them, because you're all nice people, are non-disease-causing mutations. But in some cases, they may be, and we don't know how to sort that out. So if you take CPVT, exercise-induced sudden death, as an example, a number of things that can be done is, as I mentioned, we can map the location of the mutations in the high-resolution structure. In collaboration with Ken Shepard in our electrical engineering department, we developed a high-throughput, very high resolution bilayer system where we take very small bilayers on a chip, 30 microns, which are 1/10 the size of our normal bilayer membrane, and take a CMOS integrated circuit, put it right up against the bilayer, and by reducing the capacitance of the bilayer and the noise, have been able to improve the resolution of the typical single-channel recordings from millisecond to nanosecond resolution. And also, we can measure multiple channels at a time. And with this approach, hope to be able to do the testing for leaky channels much faster in a larger number of patients. In rare instances, we could make a knock-in mouse with CRISPR, although obviously we're not going to do a lot of those. I heard some wonderful data today from Nate and from other people about iPS cells, and obviously they can theoretically be used to develop cell-based assays and drug testing. And we have generated a database with over 2,000 individual patients and almost 1,000 mutations for RyR1, which we're about to put on the internet live. And what that contains is the genetic, the mutation, the phenotype of the patient, any data that are available about the channel parameters, the location of the mutation in the high-resolution structure. And what we're experimenting with now is to try to take all of this information, first of all, make it widely available, invite people to send in more data, and try to figure out how we can use this information to tell physicians whether or not their patient's mutation is likely to be disease-causing or not. So again, this is functional genomics, and I think it's really critical to do that for the whole precision medicine. And finally, my fellow Alice Kushner, who spearheaded this and got 4 Columbia undergraduates to read over 900 papers in 1 year, every paper on the Ryanodine receptor, and enter all the data. And in exchange for that, they got a letter from me from medical school. So I think it was a good trade-off. They did a wonderful job. And Alex came up with this term here, cryo-EM structure for analyzing variants based on localization, and he calls that crystal ball. So he's a very creative guy, a little bit crazy. But okay, so I want to finish up and just mention people that have done a lot of the work, former members of the lab. A lot of you know Stephen Marks. The structural biology team was a collaboration with Joachim Frank and Amine de George in Joachim's lab, Wayne Hendrickson, Oliver Clark in Wayne's lab, and Ron Zock in my lab. And after we had published the structure and then a follow-up paper on gating of the channel, then Joachim won the Nobel Prize, and so I personally take some credit for that. So I just want to finish up by mentioning that this is a pregnant female bull shark that I photographed in Fiji. It's a 15-foot, very aggressive shark, and she came right up to my camera dome. This is a pygmy seahorse that I photographed in Bali, and it's less than half a centimeter. And in this case, the women in the room will be happy to know that it's a male and it's carrying the eggs here. These have absolutely nothing to do with my talk other than they both have Ryanodine receptors. And so thanks very much for your attention, and be glad to take questions.